Hey y'all, how you doing? This is Dr. B. This is video one of restriction endonucleases. The second video is going to be on a technique that we use them for. So let's talk about first like what they are. Let's go by the name. So the word restriction. These enzymes were discovered in bacteria in the 1950s, and basically the bacteria use them to inactivate and cut viral DNA from viruses that attack bacteria called bacteriophages. So we just basically are exploiting these enzymes that bacteria have produced, and we use them for a lot of different things. So where the word restriction comes from is it basically restricts viral activity in a host bacteria by destroying the DNA. The word endo means inside, and so you can see that these enzymes, just by the picture of the scissors here, they cut inside the DNA as compared to like chomping on the end, like if you know the old school game Pac-Man. An enzyme that would attack from the outside is called an exonuclease. So that's where the word endo comes from. And then nuclease refers to any enzyme that destroys nucleotides, or I should say axon nucleotides, or cuts them. So that's where the name comes from. It's enzymes that cut nucleic acid internally, and they were designed to restrict viral activity. So this is just a random one here, and you can see that it has a name called EcoR1. Part of the name for these comes from the organism they were found, and this was a strain of E. coli, which is the ECO, where it was found. And this enzyme, every time it sees specifically GAATTC, and you'll notice, by the way, that it's palindromic, which means it's the same on the other strand of DNA. It's the same thing backwards, GAATTC. They don't all work like that, but many do. That when it finds that particular sequence, it cuts the DNA just like this, and once it's done, it looks like that. And this creates something called a sticky end. So this is what normally the enzyme would do. So if this was viral DNA, that gene is now inactive, and in all likelihood it's going to not allow the virus to make more viruses within the machinery of the bacteria. But we can use it for all kinds of stuff. And so let's look at a couple others. So here's just some random other ones I picked up off the internet. You'll see that here's EcoR1, and it cuts that staggered kind of cut. A couple other ones like HIN3, BAMH1, they do the same thing. But then you'll see there's a couple other enzymes that just cut linearly. There is no sticky end. They're called blunt. So anytime, for example, like HAE3 sees GGCC, which again is palindromic, it'll cut right between there. And so that is the generic action of what they do. It's an enzyme that scans the DNA, and when it finds its complement, cuts the DNA. So how can we use them? Well, there's a lot of applications. I'm just going to go over the two main ones that would apply to bioinformatics and molecular biology. And this has to do a little bit with NCBI, and I'll talk about its relationship later. So basically, let's just say that you have some kind of target gene that you want to put into a plasmid. Now remember, a plasmid is a extra chromosomal circular piece of DNA that some bacteria have. They don't need them, and we can use them to do all kinds of things, like have bacteria make proteins for us. So let's just say you wanted to make or have bacteria make a human protein because you wanted to study it outside of the human body. So what you could do is you could find, let's just say this is your target gene, and hopefully somewhere outside of that target gene is a site for EcoR1, and of course there'd have to be two of them. So here's just, and again, they don't all have this, but in this case, let's just say there's a target gene, and somewhere on the upstream left and the downstream right, you notice that it has an EcoR1 site on both of them. And also what we can do is we can buy plasmids, and I'm going to show you a plasmid map near the end of this video, but we can buy plasmids that come with all kinds of restriction sites that the company put in there. And so what we're going to try to do is take this target gene, and we're going to try to kind of glue it into this plasmid, and we're going to do it using restriction sites. So basically the first thing we do on that is we then use the enzyme, And you'll see that on using EcoR1, it's going to create that sticky end on both of these, and there in the middle is your gene. And we also use EcoR1 on the plasmid that has one restriction site for EcoR1. I think you're going to see what's going to happen here. We can kind of slide this thing down there. So then what do? We basically put them in a tube together, and because of hydrogen bonding, they tend to kind of come together, at least some of them do, in this kind of manner right here. And so it's not quite done, because what we have to do at this point is we also have to use another enzyme called ligase. And ligase is going to kind of, you can see the difference between here and here, right? The backbone of the DNA is not connected. That A and G is not connected. So we use an enzyme called ligase, and that kind of glues in the backbone. And so now if you look at what we had to start with, we had a plasmid with just a restriction site, and now we have a plasmid that actually has the gene in it. And so what we can then do is we can then input this plasmid via transformation into bacteria, and bacteria will literally make this protein if it's designed the right way. And we use all kinds of genes to do that. So let me give you some examples of that. So this is a little bit of a sidestep, but I just wanted to kind of show you what a lot of these plasmids look like that you can buy. And this looks pretty crazy, but you don't need to worry about really much of anything in here, except you'll see that all these weird things like XBAL and PFOL and all those things all the way around, those are restriction sites that the company that designed this plasmid put into this plasmid so that you could have a really good chance of finding some particular restriction site that would apply to whatever gene you are trying to incorporate. And you know, there's places where they can be put in, but I don't want to get into all that stuff yet. So right now, this is kind of what the maps look like. And so you can see that if you don't happen to have an EcoR1 downstream and upstream restriction site of your target gene, you probably have one of these 58. And that's kind of the notion of generally how some of them work. There's other ones that pack them all into one very small site, called a multiple cloning site, but I won't get into all that. So that's the idea, is that using this shell and putting the gene into it and then putting this plasmid into the bacteria, and then the bacteria will end up producing whatever that protein is that you want in many cases. So how does that notion of putting a gene into a plasmid with restriction enzymes matter? Well, there's ways we do it. It's not always straight up the entire gene. And if you recall from chapter 1, if I were to put like a human gene, the whole actual DNA, into bacteria, they're not going to make what they're supposed to make, because, as you know, we take out when we transcribe DNA into RNA, we remove the introns and we just have exonic DNA. So if you were to try to put a human protein, or at least the code for it, into a plasmid to have the bacteria make it, you would need to use the mRNA code. And that's why a lot of times when you guys are going to be looking into NCBI, you're going to notice this word here, which is CDS. And basically what that tells you, you can see right here, is in many times it's just the mRNA. So the bacteria, remember, they're going to make every single letter of the DNA. And so that's why it's a great way, if you were actually going to put some sort of gene, the version of the gene that you would ligate into a plasmid and put into bacteria is going to be the CDS, or is going to be the messenger RNA. And so here is the messenger RNA for it. And if you were to compare this sequence to the entire gene that is in the cells of the mouse, that gene would be a lot longer than this, because it would include all of the introns. So now you have an idea of what CDS means. Sometimes they get this information from plasmids, sometimes they don't. So I just wanted to kind of give you a little heads up on that. So there's one more major application, and I'm going to first sidestep real quick and have you guys watch a video on gel electrophoresis, because the idea of restriction enzyme mapping, it's an important thing for you to know, requires your understanding of electrophoresis. So it's a short video, so go ahead and watch that in the Edpuzzle, and then go ahead and watch the restriction enzyme mapping Edpuzzle after that. All right, bye.